Good morning, everyone. Can you hear my voice okay?
Yeah.
Great. How is everybody doing today? Well?
Yep.
Very good. Any questions before we begin? Or comments? I had there was a question on the chat earlier before everyone joined about the midterm. If there was any information, and there isn't any information at the present time. I haven't started actually thinking about the midterm yet. So as soon as I do have some information for you, I will post it on D2L. Okay, so you don't need to worry about that.
I have one quick question.
Go ahead.
I'm hearing from some profs that they're being pressured to move classes in person. Will this one still stay online in March?
Yes, Catherine. I'm trying to keep it the way it is. Last semester, I had courses that were remote. Then they asked us to switch to hybrid, and then of course the things with COVID-19 were in flux, so we kept switching back and forth, and that was very disrupting and very annoying. So I'm trying to keep it the way it is. They are trying to encourage people to come back to campus, but I let them know that the students already plan to be away from the fall time. They plan to be away for the winter semester because that's how things looked like back in the fall, and they don't want to take a chance of coming back to campus with apartments and rent and so forth, and then after 3-4 weeks the government asked to lock down again, right? So I'm trying to keep it the way it is. Hopefully I'll win that fight.
Much appreciated. Thank you.
Yeah, you're welcome. I know it can be quite annoying to switch back and forth, and not everyone is able to make it in person. This way, the board is even for all the students. They're all getting the same delivery, and it's more consistent. I think it's more fair this way. Right. Anything else?
I just was going to say I have to TA a lab right after this class, so I might leave like a few minutes early.
No problem, Chris. It's today actually. It's not a long chapter. I believe there are only about 50 slides, so should be able to finish earlier than usual.
Awesome, thank you.
All righty. Okay, so let's get started then. Today we will be covering chapter 14, and let me. What's happening here? Oh. Okay, let's do it this way. Okay, we're good. You can see my screen.
Yep.
All right. So today we will move to a different topic. We'll start the conversation about the immune system that we have in place. The entire system is quite complex, of course. You know, it's a field in itself, immunology. So what we will cover in this chapter are the basics of the innate immune response. So this is the immune system can be separated into two distinct arms of defense. There is the innate immune response, aka non-specific immune response, and there is the adaptive, aka the specific immune response. The terminology used here, difference between non-specific or innate and specific or adaptive, refers to the fact that the specific immune response involves the activity of B cells and T cells, whereby the reaction is specific against antigens of a specific microorganism. And that's where we develop memory cells, and that's really that's why vaccines work for us. The non-specific one, as the name implies, is comprised of physical barriers and chemical barriers, and we will cover those in this chapter. And these are there regardless of the nature of the microorganism. So it will defend against any foreign microorganism, and that defense is not specific against an epitope or an antigen of a specific microorganism. Okay, that's the separation of those two types of immune responses. The field of immunology, one can say that it began with the very important work of Ilya Mechnikov, the Russian scientists who were working with a variety of organisms, including starfish and other fleas, water fleas. So essentially, Ilya was looking at using microscopic techniques to look at detailed anatomy of these organisms, and during those studies, was able to recognize that there are these type of cells, a lot like an amoeba, because of the fact that they are motile and they're able to engulf smaller organisms. So they feed on smaller organisms, as you know, in the case of the amoeba. So these cells were seen to move about and engulf foreign particles. And the same was observed for cells of water fleas, whereby these cells are able to engulf and destroy yeast cells. So together, these cells were dubbed phagocytes from phagocytosis, or the ability to eat. And of course, this very important work was published back in the 19th century, in 1884. And for that work, Ilya received the Nobel Prize in 1908 for studies of immunity. Right, now you all have background in this area. So some of it will be new. And if we get to that point, when we get to that point, and you have questions and those, please interrupt me at any point, at any time, to ask your questions. Right. So as I was explaining earlier, the non-specific or the innate immunity is present there all the time from birth, whereas adaptive or specific immunity, which involves, of course, activation of this particular part of the immune system by foreign antigens that are produced or introduced into the body. An antigen I assume you know the definition of an antigen. The antibody, you know what it is. The antigen is the reason why antibodies are produced. So an antigen is any molecule that can stimulate production of antibodies. So the anti part of antigen refers to antibodies, and gens refer to fragments or proteins that can generate the antibodies. So antibody generation. The adaptive immune response is not there at birth, but rather develops during the early years of life. And it depends, of course, on bone marrow, as well as the thyroid during development. And typically by the age of 10 or 12 years old, the system is fully developed.
Quick question.
Yeah.
Because I know when babies are born, often they have, at least for a few months, a little bit of immunity to some things passed on from the mother.
Correct.
Potentially, depending on vaccines and other things.
Correct.
That would be considered adaptive, though, right? Because it's not, because it is specific.
It is adaptive, but there are different types of adaptive responses. So there is the passive adaptive response, which is this particular one that you're referring to. It's
Okay.
It's passive because it is not actually developed within the body of the subject. But it is true. So the mother can actually deliver antibodies through the blood during the feeding of the fetus, and also during breastfeeding. It can be found in breast milk. So the antibodies, obviously, from the mom were generated through the adaptive immune response, but they were not actually produced in the body of the child. So it's referred to as a passive adaptive response.
Thank you.
Okay. Is that good?
Yes.
Okay. Right. And now, when we talk about the adaptive immune response, of course, it is the more complex component of the immune system. And because of the fact that it can attack specific antigens, which are, so an antigen, again, let me go back to what antigens are. Aside from the definition, what makes a good antigen is a protein. What makes a poor antigen is LPS, the lipopolysaccharide, or the endotoxin that is in the outer layer of, say, gram-negative bacteria, E. coli, right? So now the reason proteins make good antigens is because they have a very distinct structure. And the antibody, the immune system can recognize. Now, the immune system does not recognize specific amino acids of a protein per se, but it recognizes a specific structure on part of a large protein. Okay, so this specific structure is typically a short sequence of amino acids on the surface of the protein. Okay, it could be from 6 to 8 amino acids long. Now, these amino acids will take on a specific conformation, and it is this particular three-dimensional shape of these amino acids that the immune system will recognize. So this is called an epitope. The larger protein that was able to generate the immune response containing this epitope is referred to then as an antigen. Okay, so any antigen has epitopes, and any epitope is either an antigen in itself or a part of an antigen. That's the reason why proteins make good antigens and lipids in general make poor antigens. Right. So the non-specific immune response can be further broken down into the first line of defense and the second line of defense. The first line of defense is essentially the physical barriers that protect our bodies from invading microorganisms. And then the second part, the second line of defense involves chemicals that are released in our system, as well as subsets of cells that aid in the defense against invading microorganisms. Okay, so if invaders breach this first line of defense, the physical barriers, which are skin and mucosal membranes, there are sentinel cells that contain specific receptors called pattern recognition receptors. And these, now, I know what you might be thinking. Okay, if a recognition involves a specific molecule and a specific receptor, then why is it not considered a specific response? And that is a valid question. Now, the thing about these particular receptors, although they engage in interaction with molecules that are foreign, the type of recognition is not specific to a microorganism. That's why they have this name, pattern recognition receptors, rather than a specific protein receptor that you can find on the surface of bacteria or virus. They recognize a pattern rather than a specific part of a protein that we can call an antigen or an epitope. So there are these cells that carry these receptors, and we'll talk more about these shortly. And then we'll talk about the complement system, which is composed of a number of proteins circulating in the blood system and within tissue fluids. Cells that are part of this system can produce and respond to cytokines and chemokines produced by other cells. These can be released as a result of physical injury to cells or due to interaction with foreign microorganisms. So some of these include interferon that is secreted during viral infection, and it has a specific protective role, which we'll also see shortly. We mentioned cells that are capable of engulfing foreign debris, microorganisms, as well as debris of our own cells after they die. Our cells after they die, of course, which is, it could be a natural process because we know that our cells have a finite lifetime, or they could be, they die as a result of an infection, okay, from toxins of microorganisms, etc. So this cell debris has to be actually removed from the system, and these cells will do this job as well as defending against foreign microorganisms. We also talk about the inflammatory response after that, and its role in the second line of defense, as well as fever. So just to represent these distinct stages, now think of the first line of defense as the physical barriers, okay, just as in a wall, in a jail cell. I don't know why they use this kind of similarity. representation. Nonetheless, it relays the necessary message. Okay, so skin and mucosal membranes and antimicrobial substances released by mucosal membranes, as in the tears and mucosal secretions. These represent a physical barrier. Then we have surveillance, sensory systems that are able to sort of trigger an alarm if foreign material is encountered. So these include the ability of the pattern recognition receptors to bind these patterned molecules on the surface of microorganisms, or even free molecules like microbial DNA or viral RNA or lipids, etc. As well as the complement system. So the complement system, although it has a defense role itself, it is capable of actually killing microorganisms. It can also be thought of as an alarm trigger, because it can alert the specific immune system, and it can aid in the activation of the specific immune response during this encounter with foreign particles. And then lastly, of course, we have the so-called effector actions, and that's where essentially cells, complement activation, phagocytic cells, the interferon response that's produced as a result of cells infected with viruses, the inflammatory response, and the fever activation represent essentially quick and decisive action, which is successful in most cases at eliminating the pathogen, and in some cases the system is not effective, as you know. Okay, so let's go through the specific components of the first line of defense, which are the physical barriers and the chemicals that are produced by this chemical barrier. So the physical barrier, essentially any part of our skin and anything that covers the parts of our body that interact with the environment, because that's where microbes will essentially, in general, attack from. They come from the outside. So any part of our body that is exposed to the environment is considered part of the physical barrier. And that includes exposed parts, so the skin. It also includes the mucosal membranes that are part of the entire digestive system, from the oral cavity, of course, to the anus. We have also the lung that's continuously exposed to air and everything else that air may contain, and also the urogenital tract that is also exposed to microorganisms that could essentially swim upwards and cause infections in the bladder and so forth. So these constitute the physical barrier. The skin itself works by the fact that it has thick stratified layers of skin that are keratinized. They're very strong, binding together with high strength, and there are also dry respirations that will leave salt. There are also release of organic acids and fatty acids on the skin. So part of the sweat and part of the oil secretions. All of these constitute defense properties of the skin. And lastly, the fact that the outermost layers of the skin are continuously shed off, removed from the skin. So any microbes that physically attach to the outer layers of the skin are then physically removed, or mechanically rather removed, by means of removing these outer layers of the skin and whatever microbes attached to them. In the case, of course, of the mucosal membranes, where there are mucus-producing cells, so this could be the nasal cavity, the oral cavity, the upper respiratory tract. You can also think of them as the gastrointestinal tract as well. So in the upper respiratory tract, we have cells that contain cilia. You may recall that from cell biology. And there are mucus-producing cells that will release significant amounts of mucus to cover the entire layer of the cells. And the movement of the cilia will continuously move the mucus upward towards the throat. And any microorganisms that came into the lungs through the breathing process end up being stuck to the mucus, and then they are released or removed from the lungs as the mucus itself is removed through the activity of the mucoscirculatory system. The digestive, respiratory, and the genitourinary tracts contain mucosal secretions with chemicals. There are mechanical ways as well of removing microorganisms. In the intestine, for example, we also have the peristalsis, which is essentially the sequential constriction and relaxation of the circular muscles around the intestine, which continuously move the food down the intestine. Yeah, that's and there's also the intestine actually also moves. Every time you move, it moves also sideways. Gives additional mechanical force to the intestines to move material out of the intestines. Now, for the chemical parts, I already touched on the respiratory secretions, or the perspiration, rather, the perspiration on the skin. And as the water evaporates, of course, it leaves salt, release of oils from the skin. Oils contain fatty acids. Fatty acids have antimicrobial effects, not so much antiviral, but certainly antibacterial. Mucosal secretions also contain lysozyme, and you recall from microbiology, lysozyme is an enzyme that degrades the peptidoglycan of bacteria, and this is found in mucosal secretions. It's found also in the tears that your eyes produce whenever they sense irritations, okay, or microbes. There are also peroxidases that are produced, which break down hydrogen peroxide and produce reactive oxygen species that have antimicrobial properties. There are also the secretions will contain lactoferrin. It's a protein that binds iron. And what does iron have to do with this whole story? Well, what we find is that for a significant number of pathogens, iron appears to be a key metabolite that it requires during the infection. We can see this actually where pathogens go through the trouble and the energy expenditure of synthesizing special chelators of iron that they release from the cell to bind iron outside of the pathogen. And then they have receptors for these chelators. So these are called exochelins in most cases. They have receptors to take up these chelators and take up the iron that is bound to them as well. So because of this, obviously nature evolved, or us and the microorganisms evolved in the same environment. And it's obvious that our system during this process has learned to actually have in place mechanisms to remove iron and reduce its availability during infections by releasing large amounts of these proteins. The system will also release antimicrobial peptides. These are fairly small proteins or peptides called the defensins. And when they come in contact with the membrane of a microorganism, they will actually punch holes and eventually lead to inactivation of the pathogen. And let's not forget about the role of the normal microbiota in this defensive process. We know that microbes in our system, particularly the GI tract, play an important role in maintaining balanced health for us. And also, they defend against incoming potential pathogens. Okay? So their mere presence there physically restricts the binding sites available, the binding sites of our GI tract available for incoming pathogens. They also would compete out pathogens by consuming nutrients, because typically they exist in larger numbers as compared to incoming pathogens. And they also produce toxic compounds themselves. For example, Cutibacterium produce fatty acids, and I mentioned earlier, fatty acids can also be part of oily secretions in our skin, and these have antibacterial properties. E. coli can synthesize colicines, and these are similar to the defensins that our body produces. So these are small proteins or peptides that also will attack other bacteria. Lactobacillus in the vagina will produce during growth organic acids that would acidify the immediate environment of the microorganisms. And acidic environment, of course, low pH itself has antimicrobial properties. It discourages the growth of microorganisms, generally speaking, for bacteria. Not so much for fungus, however. As we already know, disruption of normal microbiota, most likely or most often due to use of antibiotics, or if the person is predisposed to having poor microbiota because of some underlying diseases. This will remove or reduce the protective role of the normal microbiota and open the gate for potential pathogens to multiply to large numbers and actually cause an infection. So we have good example, of course, and this is a continuous problem even today in hospitals, infections with C. difficile and Candida albicans in the vagina. Cells of the immune system. This is part of how cells of both the adaptive and the innate responses are formed. As you know, of course, all cells in the blood, red blood cells and white blood cells, the leukocytes, develop from the bone marrow. This is a process that is called hematopoiesis. In the bone marrow, we have stem cells, pluripotent stem cells that are capable of multiplying for a very large number of cell divisions. And they typically last with us for our lifetime, although their numbers reduce with aging. Okay? So these hematopoietic stem cells capable of multiplying, and they're also capable of differentiating to produce different types of cells. And these are, in fact, being considered today in research to try and rebuild damaged tissue, because potentially these can differentiate into any type of cell. A blood cell, stem cells can differentiate into neurons, stem cells can differentiate into epithelial cells, endothelial cells lining the blood vessels, and so forth. So these cells are induced to develop by hormones such as hormone-stimulating factors. And let me go directly, actually, to the schematic that describes this process. So here, these cells will undergo self-renewal, multiply or divide to produce identical cells. And then if they're exposed to hormones such as hormone-stimulating factors, they will differentiate into one of two cell types, okay, or ancestral cell types. Okay, there's the myeloid arm of differentiation, and there's the lymphoid arm. The myeloid arm, as you can see, will differentiate further to give rise to RBCs, platelets that originates from megakaryocytes, mast cells that are involved, will degranulate and involved in allergic reactions. Then granulocytes, as well as monocytes. The granulocytes, named so because of the granules, microscopically visible granules that are in these cells. They have different names because of the fact that they stain differently with cellular stains that are used during microscopic examination. And then the monocytes. The monocytes can differentiate further into either macrophages or dendritic cells. Okay. The macrophages are the cells that will engulf foreign particles and are found in the circulatory system, as well as within tissues and all fluids of the body. Dendritic cells typically reside within deeper layers of the skin, but they can also move out of the skin during the immune response. The lymphoid arm of differentiation gives rise to natural killer cells, and then both T and B cells. And so these last two types of cells are the key players in the adaptive or the specific immune response. The granulocytes, as I mentioned, those three different types. The neutrophils, basophils, and eosinophils. So upon activation, they will degranulate, essentially release the granules as they fuse with the cytoplasmic membrane, and they release the granules to the outside. So the chemicals, of course, granules contain chemicals, enzymes, antimicrobial agents, such as defensins. And we find that their numbers quickly increase during infection. So when a physician sends you to do blood work, if you're not feeling well and they suspect an infection, they would typically ask for blood count. So that includes red blood cells and white blood cells. The basophils are involved in inflammation and allergic reactions. Their granules contain also histamine. Mast cells are similar, of course, to basophils, and they're found in tissue. And then we have the eosinophils. These appear to be particularly relevant during parasitic infections, not viral or bacterial or fungal, specifically parasitic worms. So large number of these cells will end up surrounding or attaching to the outside of the worm. They activate and they release these granules, which have, of course, digestive enzymes and defensive peptides to try and neutralize the worm. The mononuclear phagocytes, they're part of mononuclear phagocyte system, and it involves, of course, different cells and organs. Monocytes, the key players that differentiate, as I mentioned, into macrophages or dendritic cells. So these are found in the circulatory system and fluids within organs. These develop outside the blood vessels, and I'll show you how. I have a video of how these cells, including neutrophils, end up leaving blood vessels, and when they are attracted to a site of an infection or a site of tissue injury. So dendritic cells are sentinel cells. They reside within deeper layers of the skin, as I mentioned, and they act as scouts, essentially. If they come in contact with foreign particles, they will, just like a macrophage, they will engulf the microorganism. It will try to break it down inside the cell, and it will try to present fragments of that microorganism to the adaptive immune system for inspection. Okay, the immune system will decide whether it deserves an actual specific immune response or not. Okay? Now, this becomes important when we talk about self-recognition, right? Because it's important for the immune system not to attack our own cells. So the way the immune system develops is such that cells of the immune system that react to our own antigens during the development of the system, they end up being deleted from the system. They killed off. Okay, so those clones of B cells and T cells that are responsive to autoantigens end up being killed off and removed from the system. Now, there are two related issues to this, and they're outside the scope of this chapter, and that involves autoimmune diseases. And we may actually get to talking about that at some point. Then we have, of course, the lymphocytes, the B cells and the T cells, and they have these names because the B cells actually develop in the bone marrow, whereas the T cells develop to their final stage in the thymus. So T for thymus. And these typically reside in lymph nodes, lymphatic tissue in general. So they circulate within lymph fluids as well. There are innate lymphoid cells, that also lack specificity, and that's why they are part of the innate immune response. But they can promote an inflammatory response, which is part of the innate immune response. And natural killer cells can also destroy. I mean, look at their name, right? They're called killer cells. So you can guess as what they can do. Okay, a very important aspect of this whole process is cell communication. Okay. These cells, there are a large variety of them in different parts of the body, and they need to be able to communicate to have an effective response, obviously. Otherwise, every type of cell would be reacting on its own, and it can create problems, which we will also see later on when we talk about a cytokine storm. Right, so communication, obviously, it involves a chemical signal and a receptor. And the signal would be sent from one cell to a responder cell, which carries the receptor. Once the receptor engages that signal, then the message will be relayed to that cell as to what is going on within the body, right? So these chemical signals then are really the cytokines and the chemokines that are released by the different types of cells. And these can be signaled to cells in the immediate vicinity of the cell that is releasing that signal, or they can, of course, have a more systematic effect because they can end up in the circulatory system and they will be distributed throughout the entire body, and they can relay that message or that signal to cells, remote cells and organs far away from the the cell that is secreting this signal, right? And some of the responses to these signals could be to grow. The cell that has the receptor and receives the signal will grow in size and multiply. It can differentiate to transform to a different type of cell. It can be a movement, or it could be a signal for the cell to actually commit apoptosis or cell death. There are also other surface molecules that play a role, actually play several roles. Their primary role is adhesion. So they attach, of course, to each other, but they're also capable of attaching to receptors that are not always there. So once a cell receives a signal, it may actually express a specific receptor for these adhesion molecules. And at that point, any molecule that contains the adhesion molecule will be grabbed, so to speak, or bound to the surface of this other cell. This becomes important when we talk about the movement of neutrophils outside blood vessels. So chemokines, again, they play a role in chemotaxis of cells. They're chemoattractants. The response of the cell here in this case is a movement, and it would typically be towards the source of these chemicals. Okay. These include colony-stimulating factors, interferons, interleukins, and tumor necrosis factor. There is TNF alpha, TNF beta, and TNF gamma. But let's keep things simple. So these have general areas of activity. The colony-stimulating factors, from the name, They would stimulate the growth and differentiation. Interferons are specific for viral infections. Interleukins are important in have general role in both the innate and the adaptive immune response, and there are a large number of these interleukins. And the tumor necrosis factors have roles specifically in inflammation and apoptosis. Now, groups of cytokines obviously can work together to generate a unique response. So a response that is different than a response that each separately will induce, okay, or generate. Here, pro-inflammatory cytokines, for example, tumor necrosis factor IL-1, interleukin-1, interleukin-6, these can, together when they're present, they will generate a distinct response than, say, TNF by itself, or IL-1 by itself, or IL-6 by itself. Now, cytokines themselves can also generate damaging responses to our own tissue. It's just a side effect, if you will. The level of cytokines is really what determines the response. Okay, so the cells are activated to release the cytokines. They would release to a specific level because of the specific number of cells and how much of these proteins they can produce. But in some cases, the response can be exaggerated or exasperated, rather, to such level where it is considered a cytokine storm. And this cytokine storm is potentially lethal because it is a system-wide response where the system will start to shut down organs, and you'll have organ failures, and that can be lethal, obviously. Okay, so before we break, let's talk about these pattern recognition receptors that we touched on earlier. So what these receptors recognize are the so-called microbe-associated molecular patterns. Okay, in some books, they're referred to as pathogen-associated molecular patterns, or PAMPs. So regardless, they are the same type of molecular patterns that are recognized by these receptors. So these include peptidoglycan or bacteria. So here the complex of the peptidoglycan is recognized as a pattern rather than specific molecules, because as you know, part of the peptidoglycan is in glucosamine, right? There's N-acetylmuramic acid and there's N-acetylglucosamine. N-acetylglucosamine itself as a molecule is present in our system, and these receptors do not respond to it. But when it is within the context of a peptidoglycan, then these receptors can be activated. Lipoteichoic acid in the cell wall of gram-positive bacteria, lipopolysaccharides in the outer membrane of gram-negative bacteria, and lipoproteins that can be released from a variety of pathogens. Subunits of flagellin proteins, and microbial and nucleic acid and viral nucleic acids. These all act as molecular patterns. Okay. There are also damage-associated molecular patterns, or DAMPs. DAMPs are produced typically inside the cell, and they are produced as the cell is experiencing damage, obviously due to an infection or due to presence of a foreign molecule, even if it's not an infection, a toxin, for example, because you can get sick by being exposed to a microbial protein, a toxin, without having an active infection, obviously. So these can activate specific toll-like receptor or intracellular receptors that can recognize these DAMPs. Obviously, the pattern recognition receptors are found on the cell surface, whereas the damage associated molecular pattern receptors are present inside the cell. Since the receptors are on the surface, these would engage obviously the molecular patterns associated with the pathogens or the microbes. Then those molecules are taken in through or trigger a signal, intracellular signal, that will eventually affect gene expression. These receptors can also be found within endosomes, which obviously start or initiate at the cell surface in any case. And the pattern recognition receptors that are free, not membrane, not integral membrane proteins as the other ones, but are rather soluble proteins present in the cytoplasm that are able to detect molecular patterns of microbes that are released within the cell. So those that escape the endosome, for example, or that can penetrate the cell membrane directly from the outside, can be picked up by these soluble pattern recognition receptors. So these pattern recognition receptors is a family. So far, I believe we are up to 12 or 14 different types of receptors, and they're named toll-like receptors. So TLR1, TLR2, TLR3, etc., etc. Each of them recognizes a specific molecular pattern. Okay? So some can recognize flagellin, others lipoproteins, LPS, lipopolysaccharides, double-stranded RNA, bacterial DNA, single-stranded RNA, and so forth. Okay. Dendritic cells have both toll-like receptors and C-type lectin receptors. Okay, and we find the C-type lectin receptors also on the surface of macrophages. And as the name indicates, the lectin receptor means it's a receptor that's able to bind a lectin, or it's a lectin itself binds polysaccharides. And we know that polysaccharides are typically on the surface of microorganisms, and themselves can be recognized and bound by these receptors, and they can in fact induce phagocytosis in these dendritic cells or in macrophages. So there are the reg-like receptors, or RLRs, that are in the cytoplasm, specific for viral RNA. Often they recognize double-stranded RNA that does not have a cap on it. This is how these molecules distinguish viral RNA from ours. We typically do not have double-stranded RNA in any case. The node-like receptors, NLRs, also soluble receptors. They are able to recognize microbial components or cell damage. In dendritic cells and macrophages, those related cells, obviously, they combine with other proteins to form an inflammasome. So within the inside of the cell, These will complex with a number of other proteins. They form a large supramolecular structure called the inflammasome, and its role is to to facilitate the secretion of interleukins. For example, because these interleukins and similar molecules are produced in the cell in an inactive form. So transcription is activated, the proteins themselves, so these are the interleukins, the tumor necrosis factors, and those are all proteins, essentially. They're synthesized on the ribosome, but they remain in the cytoplasm in an inactive form. Formation of the inflammasome, in this case, will actually bind these inactive proteins and activate them before they are being released by the cell. Okay, so that is all for the TLRs. Now let's stop here at the interferon response. Let's take a 5 and rejoin to continue. We're about halfway through the chapter, by the way. Okay, everyone, let's let us continue. Right, the interferon response, a very elegant system. Okay, so follow me with the schematic to the right. Okay, so in in cell 1 here, we have a typical infection of a cell with a virus. And the sequence is just like what we've described in the previous chapter. Now there are sensors in the skin that got infected. The cell will release interferons. The interferons are will diffuse, of course, to neighboring cells. Cells with their receptors will engage the interferon, and that will activate genes for inactive antiviral proteins. Okay. Now, the initial cell 1, of course, it's too late for that cell. It has been infected, the virus multiplies, and its fate is pretty much sealed eventually. The virus is released, and now it will diffuse also out to neighboring cells to infect them. The inactive antiviral proteins are not effective in stopping the virus from invading the cell, but it has a role. Once the virus actually releases its RNA, these proteins are activated, and basically they will induce programmed cell death. Okay. There are also proteins that are able to attack and degrade the viral RNA, right? Thereby actually making a very effective strategy to limit the infection by the virus to a very small area within the tissue, and allow the immune system, of course, to eventually attack the virus and remove it from the system. This is not foolproof process. There are viruses that can escape this defensive mechanism, unfortunately. Next, we go into the complement system. As I mentioned earlier, these are a number of proteins circulating in the blood and tissue fluids. And what I'm going to do actually, so there's this slide, and this slide, 3 slides. I'm going to skip by showing you a video. What's happening there? Are you guys still there?
Yeah.
Can you see the video?
No, we
No.
see you just for now.
Just me? Where'd they go? How about now?
Nope, still just see you.
Okay, let me try something else. Where is my share screen? Okay, here we go. And I want. How come it doesn't show? How about now?
We
You're
see your
sharing
Zoom.
the Zoom window.
Oh, you just see me?
We see you and we see you sharing all the screen.
You see the video?
You see
We
your
see your
email.
You do?
Yeah, we see your emails.
Can you hear that? From the video?
No. No, we can't hear any videos. We just see your emails.
Oh, my emails. Oh, you don't want my emails. Okay, I don't know why the sharing is so complicated. Let me see. New share. How about now?
Yep, can see it now.
Yeah,
Oh,
there's the
right.
video.
Yeah. Good.
I don't think we can hear anything from it though.
What's that?
I don't think there's any volume. There's no sound to the video unless it's playing.
Is it too low?
I can't hear anything. I don't know if anyone else can.
I can't either.
It might be because you have your headphones plugged in, Professor.
Okay, let me take them out.
Nope.
Still no sound, yeah.
I think the sound is turned off for the video. When the controls popped up last time, there was the X.
Sorry, guys. I don't know why the share does not include volume.
Could you put subtitles on instead?
Let me see. I'm not sure it actually, this player allows me to do that. Oh, hold on. Oh boy. Okay, you know what I'm going to do? I'm going to just upload the video onto D2L for you to watch at some other point, okay? Just to help you actually put the picture together,
That works.
okay? So maybe what I'll try and do, actually, I'll try to use my voice to describe the video. Okay. Let's see how that one pans out. Okay. Right. So you've got the proteins that essentially lead to inflammation, cell lysis, and opsonization. As you can see, number of proteins, they're named C for complement, from 1 up to 12. There are 3 different pathways to activate the system. There is the classical pathway, the alternative pathway, and the lectin pathway. Okay. So activation starts with binding of complement component 1 to antibodies. The antibodies would typically have been generated previously. So here, this particular branch of the complement system is activated subsequent to a primary exposure to the pathogen, where your body would have generated antibody during the first encounter with this pathogen. Okay, second encounter, you'll have the antibodies binding to an antigen on the surface of the bacteria. Complement component 1 binds, then complement component 2 bind. 2 complement component has 2 fragments, C2A, and complement component 2B, C2A and C2B. This complex is called C4 convertase, which converts complement component 4 into C4A and C4B. So a convertase essentially is a protease. Okay. The protease will cleave complement component 4 into those 2 fragments, A and B, and each one would have a different role to play in the process. Okay, one of these is formation of another convertase called C3 convertase. Okay. C3 convertase will cleave complement component 3 again into 2 fragments, complement component 3A and complement component 3B. These also have different roles to play. C3b will associate with an antigen then it will react with other blood proteins to form convertase, C3 convertase, again, cleaving C3a and C3b. C3a has a role in inflammation. Okay, it stimulates mast cells to release histamines and also attracting phagocytes. C3b, on the other hand, has a role in opsonization. So opsonization is a process of specific targeting of bacteria for phagocytosis. Okay. So macrophages or other phagocytes are able to bind C3b that is bound to the surface of a pathogen. And this, of course, triggers the engulfment of the pathogen. So in this case, the marking of the bacteria by C3b and then the recognition of C3b by receptors on the surface of the phagocyte to induce phagocytosis is called opsonization. And opsonization can be mediated also, by the way, by antibodies directly without the involvement of complement proteins. Here we have engulfment. And you know what happens after engulfment in phagocytosis. The other component forms another convertase called C5, which eventually breaks down C5 into C5A and C5B in the same mode as the other convertases. C5A is involved in inflammation again, whereas C5B will remain associated with the surface of the pathogen. It will then recruit C6, complement component 6, 7, 8, and multiple copies of complement component 9. The multiple copies of complement component 9 essentially associate with each other in a self-assembly mode to form a pore. They punch a hole in the membrane of the pathogen, of course, causing the release of metabolites. This is called the membrane attack complex, or MAC, which eventually would lead to killing of the cell. Alrighty. Right. Was that effective, or?
Yeah, that was good.
All right.
I hope you uploaded because it was a lot of information, and I would appreciate if I can go back and rewatch it.
Oh, for sure, I will. I will. Okay, so we're done with the complement component. The alternative pathway is activated with C3b, binding directly to the surface of the bacteria. The lectin pathway. This is mannose binding lectins, or MBLs. So this is a protein that's also circulating in the blood. Any mannose residues in the surface of a pathogen will attract this protein, and that will activate the complement system. And we have the classical pathway where we have antibodies that actually recruit complement components. These are the three pathways shown here. Inflammation, inflammatory response, and then formation of the membrane attack complex. Here you have very nice electron micrograph image showing the individual pores that are produced by the membrane attack complex and the membrane. Okay, that's still complement. Okay, right on. So let's move on to phagocytosis then. Right, so macrophages that are attracted to a site, or even if encountering a microorganism or any other foreign particle during its movement within the circulatory system or within tissue fluids, of course, they will bind the microorganism. This could be direct binding. It could be mediated by an opsonine, like such as the complement components or an antibodies. That will trigger the formation of the pseudopod around the microorganism. Eventually, the pseudopods would meet around the entire microorganism, forming enclosing it completely, and then taking it inside the cell in the form of a phagosome. Okay. So instead of an endosome that contains molecules, a phagosome contains an actual particle, such as a bacteria. Now, of course, the whole idea of doing phagocytosis, as you know, is really to neutralize the microorganism. Okay. So here we have activation or opsonization of a microorganism with the complement component B, C3b. Then binding of the complement component will take up the bacteria eventually into a phagosome. The phagosome fuses with lysosomes that are produced that originate, of course, in the endoplasmic reticulum. They fuse with the phagosome to form the phagolysosome, and this causes, of course, acidification because the lumen of the lysosome is acidic, and also release of digestive enzymes that end up attacking the microorganism. And the idea, of course, is to neutralize the microorganism completely. And any material that cannot be reabsorbed through the membrane of the phagolysosome are released through exocytosis to the outside. So macrophages are always there, right? They do the job of a cop on the beat. They are capable of recycling our own cells, right? Debris of our own cells, as well as microorganisms, the invading microbes. They live for weeks or months. Their lysosomes are able, of course, to regenerate. Macrophages are activated in response to cytokines, which are released again by other cells, including damaged cells. M1 macrophages have very, very good killing power. M2 macrophages have a role in actually reducing the inflammatory response. If insufficient, they can fuse to form giant cells. Macrophages can fuse to form giant cells. Then macrophages, giant cells, and T cells, we find them in granulomas. Now, granulomas, I don't know how many of you are familiar with this structure, but if there is a site of infection, okay, whereby the body is unable to completely remove the microorganism and recover the inflammatory response from the site, so this would be the normal situation. Tissue reverts back to normal tissue, and the microorganism is gone, and there's no more inflammation. Okay? If this is not possible, and this can happen, what happens is the body, the macrophages, the giant cells, and the T cells that have been activated during the immune response will end up staying in the immediate vicinity of the site of the infection. Then additional cells will actually secrete fibrous material, extracellular fibrous material around them, as well as other cells that surround the site to contain the site until the material inside this structure is free of microorganisms. And what we find inside is the cell debris from our own cells, the cell debris from the pathogen, those that have been destroyed by our own cells, and the material that's secreted by these cells together, they form a gel-like material inside this structure that we call granulome. Okay, please do look up that, Google it or on Wikipedia to get to know that structure better. And so this structure stays for long periods of time, and in fact these can be visible using X-rays in patients with tuberculosis, for example, because the microorganism is difficult to remove. We find that it's common to see the formation of these granulomas in the lungs, and these can be visible with X-rays. And in fact, it's one of the diagnostic signs of tuberculosis. The neutrophils, on the other hand, acting as the SWAT team, neutrophils are very aggressive. They're more aggressive than macrophages or dendritic cells in attacking microbes. They're very fast responders, and they move quite readily throughout the body, and they can go through, they can penetrate the wall of capillaries and blood vessels to move out into adjacent tissues. So they play a very important role, of course, in early stages of inflammation because of these capabilities. Once they're very short lifespans, 1 to 2 days, and they also die once the granules they have are released. Okay, so they kill microbes again by phagocytosis and also by release of granule content. Can release DNA from to form neutrophil extracellular traps, okay, to catch microbes. It's a very interesting phenomenon. DNA, because of its inherent biophysical properties, being a polymer, it has some hydrophobic properties, and it also has hydrophilic properties, right? Because it has phosphate backbone, it's negatively charged. It seems to be an attractive site for microbes to attach to. Aside, of course, from the fact that microbes are capable of taking up DNA directly from the environment, they can also use it for attachment. So these cells can produce these extracellular traps, allowing enzymes and peptides in the granules, of course, to destroy the microbes once they attach to them. And the inflammatory response happens, of course, it's a very important response, although it's bothersome for some individuals for minor injuries and minor infections. Of course, a skin pimple is an inflammatory response in itself, a very localized inflammatory response. But we can have internal inflammatory responses, again, due to an infection or an internal injury, by the way. The purpose of the inflammatory response, of course, is to contain the damage, localize the response, eliminate the invader, and restore tissue function back to normality. During this process, you recognize the typical signs of inflammation. There's swelling, redness, increased sensitivity, pain, heat, due to, of course, increased circulation of blood on that site, and sometimes loss of function. A particular area becomes obviously non-functional. So pattern recognition receptors obviously can trigger an immune response. So cells that have these receptors will release inflammatory mediators such as cytokines and histamines, and these will act as chemoattractants for neutrophils and other cell types, as well as, of course, mediate tissue repair and attracting other types of cells, and antimicrobial, of course, defense mechanisms. Now, inducers of these obviously includes the microbes. Are also damaged tissue. Our own damaged tissue can induce an inflammatory response, as I mentioned. So, microbes-associated molecular patterns can release, of course, cause the release of tumor necrosis factor, as we mentioned, which induces the liver to produce a protein, a set of proteins called acute phase proteins. That can participate in the activation of complement. And these, in fact, in some blood work, the physician may request quantification of acute phase proteins in the blood, again, as an indirect indication of an infection. Blood vessel damage can start also through enzymatic cascades, which lead to coagulation to, of course, contain the loss of blood from the blood vessel, but also increase vessel permeability to allow for neutrophils to penetrate and across the wall of the blood vessel to reach tissue where the pathogen might be. So the vascular changes, and this for the inflammation, this and a few slides. I also believe I have a video. So I'll upload the video for this one as well. Okay. Right. So I'll go through the schematic to shortcut the wording in the slides. Okay. So here in this scenario, we have sharp objects compromising the skin, causing damage, and along with this damage of tissue, there are, of course, microorganisms that have been brought in as a result of this, okay, by microbes. Now, we mentioned in the skin, we have resident phagocytic cells, such as dendritic cells, as well as macrophages that will recognize, of course, the microorganisms. They will take them up through phagocytosis, and the damaged cells from the injury will also release pro-inflammatory cytokines, and that will cause increased blood flow. It will cause vasodilation, and the vasodilation will also work with the chemoattracting properties of these chemicals to attract monocytes and neutrophils to the site. Then there is the process of diapedesis. Diapedesis is whereby the cell actually penetrates the wall of the blood vessel. There is also, because of the increased dilation of the blood vessel, there is also loss of fluid. So we have accumulation of fluid, which we've mentioned is part of the inflammatory response. These chemicals also can induce pain in neurons in the immediate vicinity of the site of injury. The neutrophils, of course, recruited macrophages will start to work on the bacteria, again, to eliminate all of them. Tissue repair also starts. Any excessive fluid is removed back into the circulatory system through the circulatory system, and the repair of the tissue starts. Eventually, the blood clot itself is dissolved away and leaving eventually a scar. Okay. So the diapedesis process starts, as I mentioned a few moments earlier. These chemicals that are released will actually trigger endothelial cells lining the capillary to express these receptors on their surface. Okay. The receptors called selectins, and there are receptors that can bind integrins on the surface of the neutrophils, okay, and monocytes. Once these are expressed, of course, so these neutrophils and monocytes that are rolling down the capillary will end up being stuck to these receptors. Then eventually they will start a process of rolling over to the side and start, as you can see here, they form very tight association with the endothelial cells, then eventually penetrate any opening in the immediate vicinity and cross the blood vessel to the outside, to the tissue to do their job. And that is briefly the process of the inflammatory process. Okay, so it is a protective process mechanism, but we mentioned also that it can cause damage, right? Enzymes and toxic compounds that are released from phagocytic cells meant to, of course, neutralize microorganisms can damage our own tissues, okay? Because even though our cells recognize our own tissue, the proteins and that they release, digestive enzymes, for example, they do not discriminate between our own molecules and foreign molecules. Okay? They will attack either molecule indiscriminately. Although the damage is normally minimal, and think of it as a side effect and an acceptable side effect, but in some situations where we're talking about the blood-brain barrier or the spinal cord, these sites cannot tolerate really extensive inflammatory responses, and this process can be damaging, even life-threatening. In situations where we have an autoimmune disease, right? Here, an autoimmune disease is a response, an immune response, which can also precipitate an inflammatory response at the same time against our own tissues. And this can be, again, due to a fault in the development of the immune system itself. So we can have there are genetic predispositions for autoimmune disease, but there are autoimmune diseases that develop with time as a result of exposure to pathogens. They can precipitate or cause autoimmune disease. Rheumatoid arthritis, as an example, can progress to the stage of actually disabling joint function. And this type of disease is not a developmental disease or a genetic disease, but rather it develops with time, with age, as a consequence of exposure to different pathogens and the development of antibodies that end up reacting with our own tissues as a result of that encounter with those specific pathogens. We know that chronic inflammation is also associated with cancers and certain types of obesities. So inflammatory response is good, but it has its drawbacks in some cases, however. Right, now cell death. There are different forms of cell death. There's so-called traumatic cell death as a result of physical injury or as a result of attack by toxins. There is cell death, self-destruction, such as apoptosis, as you know. Apoptosis itself, it does not trigger an inflammatory response because it is a normal part of recycling of our own cells. So it typically does not trigger an immune response. Pyroptosis and necroptosis are types of cell deaths that definitely trigger an immune response and sacrifices infected cells. So this, in the case of, for example, a virus-infected cell, it may undergo this type of cell death and trigger an inflammatory response as a protective step. Okay, lastly, we have the fever, right? An agent that triggers increase in the core temperature of our body is referred to as a pyrogen. We have indigenous pyrogens, so those are cytokines that we release to trigger fever, and there are exogenous pyrogens that come from the outside. An example is the endotoxin of gram-negative bacteria and E. coli, specifically the LPS, the lipopolysaccharide. Any temperature, oral temperature above 37.8 degrees is regarded as fever if you're using, of course, the oral thermometer. Now, why fever? I know fever is associated with discomfort, typically during infections, right? But there are, in fact, reasons for our body to trigger fever formation. We find in pathogens that they are sensitive to increased temperature. Even an increase of only 2 degrees Celsius can affect bacterial growth. Moderate temperature also, as you know, increases rate of enzyme activity, and we depend on enzyme activities during the response, during the immune response, right? Enzymes are required, of course, to synthesize cytokines and chemokines, protein synthesis, metabolic activity, toxin activity, antibacterial peptides defenses that we make. All of these activities are stimulated by increased temperatures. So it's supposed to have this beneficial effect, right? And that, ladies and gentlemen, brings us to the conclusion of the innate immune response. And I'll be happy to take any questions if you have at this point.
What was the name of the pyrogens? Not the indigenous ones, but the ones that come from E. coli, for example?
Oh, the endotoxin is a good pyrogen, or not a good one in the sense of good and bad, but a good trigger of fever. It's the LPS, the lipopolysaccharide, or the endotoxin, aka endotoxin, right? That one can trigger fever.
Thanks.
You're welcome. Anybody else? All right. Well, as I promised, there's a short chapter. Thank you for your attention. Thank you, everyone. Thank you, Teresa. Have yourselves a great afternoon. Stay warm and stay safe, and we'll see you next week.
See you next week.
Bye.